è andata, meno male che il ragazzo prima mi sta a posto, perché abbiamo adesso l'onore di invitare Samuelson dell'Università dell'UTA. È un fisico, non è un medico, ma spesso gli avanzamenti della medicina sono legati ai chimici, ai fisici, ai bioingegneri. Ebbene, Gary Samuelson, che è il padre di questo argomento così interessante, così utile anche ai fini proprio della salute delle cellule, questo delle molecole di segnalazione redox, quindi ho il piacere di invitarlo sul podio, please.
Grazie e buongiorno. That's about all I can say. I thank very much the organizers of this conference for this excellent opportunity to stand here in Rome, where we have thousands of years of developmental knowledge in medicine, and it's a great honor, and I'm grateful also to be able to present to you some ideas on oxidative medicine and how oxidative stress and other things work. First of all, I wanted to start off by dispelling a myth that has been around for quite a while. The antioxidants that we say, and I just say this in passing, they say come from foods and other things that are important for us and are ingested, and that the antioxidants come from foods, and the oxidants then are these free radicals that are terrible for our health. This is not true. On a microbiological basis, the antioxidants and oxidants in the microcellular environment are produced by the mitochondria in oxidants and by the cellular, the oxidants are produced most in mitochondrial metabolism, and the antioxidants are produced inside the cells. They're the most important factors in the balance of oxidative stress in our cells, and they're companions. One cannot live without the other. Now, I'm an atomic physicist, so I'm going to talk a little bit more about the chemical and atomic properties of oxygen. Oxygen is a free radical. It has unpaired electrons. This is how I look at an oxygen molecule. If we were to then look at the mitochondrial processes, about 90% of the oxygen and nutrients and hydration are processed by the mitochondria. The mitochondria produces the energy, the ATP, that's necessary to fuel all the intercellular processes, but it's also producing superoxides and hydrogen peroxide and hypochlorites. These are the reactive oxygen species that we're talking about today. Before we thought that these intercellular reactive oxygen species were terrible, that they were damaging to the system. But we've found now through research that without the intercellular reactive oxygen species, we would not be able to have an important communication system. It's a fundamental communication system inside of our cells. It forms a network of communication that allows the cell to be able to detect, repair, and replace damaged tissue. The oxygen-free radicals, reactive oxygen species, are such that oxygen, if it receives an electron, oxygen molecule, if it is reduced, it receives an electron and turns into a superoxide anion. Then if it receives another electron, it turns into a peroxide, like hydrogen peroxide. It is reduced further then into hydroxyl free radicals, And the last, most reduced form of oxygen is water. This is called a redox process, or it's a reduction oxidation process. And the reduction and oxidation of oxygen is a very important process in cellular biology. As we know, photosynthesis takes the energy from the light that is produced. It goes in reverse, and from superoxides and such produces oxygen. And our own metabolic metabolism then takes the oxygen and does the reverse process and reduces it again to water. It's a very basic process. Inside the mitochondria during our metabolism, these reactive oxygen species are used as regulatory mechanisms. Here we see in the bilayer of the mitochondria the processes of mitochondrial respiration. And superoxides and hydrogen peroxide are produced by these processes, which are introduced into the inner mitochondrial cavity and form important functions to be able to regulate our mitochondrial function, or I'll go on and explain this a little bit. For example, hydrogen peroxide, H2O2, is able to dissolve the hydrogen bonds in thioredoxin and produce a conformal change in thioredoxin. This conformal change then allows the thioredoxin to act differently inside the cells. Thioredoxin reductase then is also employed then to return then the thioredoxin to its regular state. So oxidation and reduction cycles actually are able to change the conformation of molecules inside of our cells. And these molecules act as semaphores, or important semaphores then that direct the metabolism of the whole cell. I'm going to show you a little bit of importance on or a picture. I'm sorry, this is from Harvard Medical School. This is an animation of the cell. It allows us to see what the cell might look like inside in normal processes. This also shows that reduction and oxidation processes are inside the cell. All of these micro mechanisms that involve the conformational changes of the molecules inside of our cell require oxidation and reduction. Oxidation then is able to release these bonds, and the oxidation then, and then the reduction is able to then reattach the bonds. For example, the formation of these microfibers here are a reductive process. And then inside the cell, when they fall apart, when they're disassembled, that's a oxidative process. This is a little kinesin, and he's stepping there about 2 million times a second. And he's delivering then proteins to all parts of the cell along these microtubules. Every little step that happens is an oxidation reduction cycle. This is the illustrator. David Belinsky, and he is the one that for Harvard University has done some of these animations for us. The mitochondria. I like to show these because medical students, when they're studying the intercellular processes, it's great to have a visual context as to what it actually is taking place inside the cell. The Golgi complexes that are delivering the proteins and sorting proteins on every part of the cell. Now notice this oxidative process will release and cause conformational change inside the molecules that attach membrane structures. This is a leukocyte. It'll be dissolved and will be able to go out of the blood flow into the place where there's high levels of oxidative stress, as we mentioned before, as was mentioned. And all of this is happening because of a reduction in oxidation processes inside the cell. If we were to then completely eliminate oxidation or the processes of reduction, then we have several problems. Now, I didn't come prepared to show you some studies because I expected a little bit different audience. But what happens, there have been studies where mice have been given high levels of intravenous antioxidants, vitamin C, L-acetic acid, in order to be able to eliminate all the oxidants inside of the system. These mice, instead of living longer and prolonging the longevity of the mice, became cancerous, diabetic, morbid, and they died early. And it was wondering, well, why in the world, when we're reducing the oxidative stress inside of the cell through oxidation, through these antioxidants, why would we be able to, we have actually reduction in longevity? What I wanted to say is that redox signaling, or the signaling between molecules, is the secret of life. The harmonious orchestration and the coordination of all of the molecules inside of our cells rely on reductive and oxidative processes. The interconnected molecular players then are orchestrated by what? By an interconnecting ocean of salt water and vital fluids. Every message that is passed between these molecules needs to go through this medium of salt water and vital fluids. The redox signaling has been studied throughout the world. It's been published in publications, in journals and in conferences worldwide. It is well known. Even James Watson has become a champion of redox signaling. And he says that the molecules called reactive oxygen species, or ROS, produced by the mitochondria, help forge the chemical bonds which stabilize the proteins as they fold, and also are a force against oxidative stress in the body. They also, through the NADPH complexes, are able to kill bacteria. This is the process of, oh my goodness, phagocytosis in the white blood cells. And these very oxidants, these reactive oxygen species then are essential in being able to kill the bacteria, and they have to be 100% effective. glutathione peroxidase inside of our cells is able to eliminate about 10 million oxidants per second. So we have some very interesting things happening inside the cells. I'm sorry. I wanted to mention a few things. So besides mediating all of the intercellular processes that happen inside the cells, the reductive and oxidative processes, it also is able to have a potent antimicrobial effect. And this was fascinating to me when I first started studying when I first started studying this redox signaling molecules and realized what an essential role they play inside the cell. It was absolutely fascinating to me that these molecules can be absolutely non-toxic, and I'll explain that a little bit later. Inside of our body. Okay, I want to go into one of my favorite molecules, the glutathione peroxidase molecule. Inside a glutathione peroxidase molecule, there are approximately 4 of the glutathione. When glutathione is reduced, then it has a thiol, which is this HS on the bottom there. And the oxidation processes then are able to take off the hydrogen and produce a sticky sulfur molecule, and then oxidized glutathione is formed. Oxidized glutathione is an indicator of oxidative stress inside the body. These type of processes then allow that the reactive oxygen species produced by the mitochondria are able to be eliminated by the cells in glutathione peroxidase and catalase and superoxide dismutase, et cetera, are enzymes that are able to eliminate these. If we look at the overall homeostatic balance of the cell, of a healthy cell, these oxidants are produced at the same time that they are eliminated by the antioxidants, and this forms a homeostatic balance that's essential for the regulation of the cell. This is a healthy cell. If a cell then becomes disturbed for whatever reason, then this balance is disturbed, and oxidative stress builds up, and an excess of reactive oxygen species. And it's a bit like a smoke alarm inside of our cells. When this condition of oxidative stress exists inside the cells, it starts to produce press the genetic buttons that allow the cell to repair and replace itself, to detect the damage and to repair and replace. Without oxidative stress, then, the cell would not be capable of repairing or detecting, repairing, or replacing these cells. So that you have three options. The cell can either repair itself when it's in oxidative stress. If it can't repair itself, then it has to undergo apoptosis. And then apoptosis then, of course, will eliminate the cell, and then it will be replaced by regular cellular mitosis with normal healthy cells. If we were to look at this underneath the microscope, we would see, this is a wonderful picture here. You can see the green represents the oxidation levels inside the cells. On the left, we see normal healthy cells. On the right, we can see cells that have been damaged by X-rays. And the cells that are highly stressed exhibit oxidative stress. This could be due to malfunction of DNA, bacterial infections, or viral insults. Whatever the case be, these oxidative stress levels are increased. These create the signals inside the body that cascade the cytokines that bring in the immune system. The immune system then, if we were to look under the microscope, this was what it would look like, is able to eliminate then the cells that have been damaged, or the unhealthy cells, and the microbes. And once the insult has been eliminated, then damaged cells are replaced by normal cellular division, simulated to fill in the gaps. This is healing on the cellular level. This is what happens. And it employs the oxidative processes in redox signaling. What if we were then to be able to have a more direct impact on these oxidative processes in the cells? We mentioned some of them that are very important. Nutrition, hydration, and these other fundamental important things in the cell. Nutritional elements are essential. Of course, hydration is essential. Are able to have the cell operate on an optimal level. But what if we were able to then produce these redox signaling messengers that help to increase the efficiency of detection, repair, and replace cycles inside the cell? And this is what I found that was possible. We were able to stabilize then a combination of redox signaling molecules using simple salt and water. Through reduction and oxidation processes and a catalytic process, we're able to produce hydrogen peroxide, oxygen, superoxides, and hypochlorites that were stable in the intercellular environment in salt water. And here are some of the chemical moieties that we were able to show that are in this substance, and the concentration levels that we found. These oxidative therapies that I have expressed, the oxidative are rampant, are very important in different types of medicine and oxidative medicine today, such as ozone therapy, photonic therapy, hydrogen peroxide, and chlorine dioxide. All of these elements produce oxidation or oxidative elements inside of our cells. Exercise also, by the way, is absolutely essential. It can produce oxidative elements inside of our cells. And these oxidative elements then are able to increase the efficiency of cellular detection, repair, and replacement of the damaged cells. One of the questions we had at the very beginning is this substance safe? It has a lot of oxidants, and I didn't even want to drink it right off the bat when we we had to do several studies. So I sent it off to a colleague. Now, I'm not a microbiologist. I'm a physicist. So we sent it off to a microbiologist in a national laboratory in the United States, and they did toxicity tests and found that even in high concentrations, up to 20%, there was absolutely zero toxicity in cellular environments. These are small cell epithelial, small lung cell epithelial. Up here represents the translocation of a subunit of NF-kappa B, which if it's toxic, you see translocates into the nucleus. When hydrogen peroxide, which is one of these oxidative elements, is introduced you can see toxicity. But when this special stabilized mixture of oxidants is introduced, we see absolutely no toxicity at all. We have 20 years of studies now. And in these 20 years of studies, we have seen absolutely no toxic response at all to the substance. Well, then I asked my friend, well, what does it do? We found that it increases then the efficiency and efficacy of the intercellular antioxidants, such as glutathione peroxidase and superoxide dismutase. And the antioxidant regulation, upregulation through NRF2 was very significant, but the efficacy of these antioxidants increased more than 400%. And at times over 1,000%, which for me was interesting. We are introducing oxidants and we're getting antioxidant then characteristics. We also did preclinical trials in which we showed 100% kill of all microbes tested, and bacteria and viruses, over 30 resistant strains in less than 30 seconds. And there was no effect on the in situ beneficial flora of the gut. We saw that there were no inflammatory markers stimulated, absolutely none in healthy individuals. And the antioxidant efficacies went up, kill rates went up in damaged cells, increased sensitivity to some hormones, and it actually reversed senescence in some of the cell cultures, which means that they became more receptive to hormonal stimulation and TNF-alpha. We saw that NRF2 activation channels were opened, that detoxification channels in the gut were opened, that antioxidant expressions were increased, that it activated stem cell adhesion and differentiation in certain processes that were key to the cellular regeneration cycles. In fact, cellular regeneration in some cases went up more than 16% in skin cells when they were exposed to these substances. What's the effect of redox signaling then in supplementation in animals? We saw that in over 20 years of data in mice, rabbits, and dogs, zero toxicity, again. Normal histopathologies in all tissues and organs. And again, these were doses, very high doses that were designed to be able to kill the animal. We did not see any lethal dose in animals. Mice ran 29% longer to exhaustion. Antioxidant capacities in these animals were increased. We saw reduced, for example, glutathione levels go up in the animals. We saw C-reactive proteins go down. Lipid peroxidation and glutathione oxidation were decreased, and the metabolic markers were normalized. Here is one of the data that shows that endurance running of these animals actually increased 29% versus a saltwater placebo. So to resume, dramatic increase in endurance of animals, increased availability of energy reserves. We think that might be because of glycogen depletion is slowed down. Increased antioxidant efficacies. There's less oxidized glutathione in master antioxidants. And there, again, we saw absolute zero toxic effects in these animals. Now, as a researcher, I'm sure that you understand that when we see absolutely nothing as far as toxicity for a substance, if it does anything, then we're winning. We have clinical studies in humans, These third-party independent studies were performed in major clinical laboratories. They were well-done studies, randomized double-blind studies. There were no adverse effects in over 160 human subjects with complete blood work done. And the metabolic results were also very interesting. We monitored about 273 metabolites. Out of those, 60 plus changed significantly when they took up 4 ounces of this substance in the morning. Lipids, fatty acids, and sugars and metabolic intermediates profiles changed. And so we were able for the first time to do pharmacokinetics. And we proved that indeed there is absorption and an active process of metabolic shifts inside of humans. Here's some of the data. I'm not going to explain exactly what this means, but these blue dots represent normal metabolite profiles. And then when introduced, again, 4 ounces of this redox signaling compound, we saw significant shifts in the metabolic profiles. Doing our PK analysis, then we were able to see that over time, these profiles shifted and changed. And up to 24 hours, there were measurable shifts in the metabolic profiles in these subjects. Now we have 25 years of in vivo toxicity research. And in those 25 years, we found that there's no endotoxicity, cytotoxicity. We found absolutely no toxicity, no reverse mutagenic problems, no genetic problems, no LD50. There's complete lack of side effects and in any form of administration, and no abnormal histopathological effects. So if anything, we knew this was safe. We knew that it was safe. Highly oxidative compounds, when they are balanced and stabilized, are able to be shown to be absolutely safe and antimicrobial inside our organism. We did tests on skin. We saw reduction in wrinkles, reduction in blotches, reduction in smoothness. All sorts of skin damage issues were addressed. We saw improvement in texture and in elasticity. Here are some of the examples then that only after 30 days or before 30 days saw significant improvements then in skin texture. We also looked at the blood flow in the skin. Blood flow increased over 10%. Decreases of cellulite and fat globules increased 16%. That was good for women. Increases in the rate of cellular regeneration then increased also by 16%. So this redox signaling network that exists inside of our cells is able to then help us, the cells, to detect the damage when there is oxidative stress condition. Activate the genes to repair and replace the cells. And thereby then we have normal cellular regeneration. When we supplement these, we start to see accelerated regeneration. Here is regeneration of a person who had severe third-degree burns from a wax burn. When these redox signaling compounds were applied only 3 times daily, no skin grafts were needed in order to replace the damaged skin. The skin grew back without scarring, and this gentleman, after 6 weeks, had full function of his hands. We've seen this over and over again. Very, very quick recovery from surgical procedures. very, very quick recovery from other degenerative problems, and also we've actually seen regeneration of tissue, internal organs also. But I won't mention that right now. Redox then, applications. We can use this to reduce oxidative stress. We can use this also to normalize immune response. When these indicators are present, it actually helps to normalize the response because the network then of cells able to communicate. Often autoimmune disorders are caused because of a lack of communication and rampant oxidative stress. And when we're able to reduce oxidative stress and increase the ability of the intercellular communication through this redox signaling process, then the immune system tends to normalize. We've seen normalization of hormonal responses, even with people who have had their thyroid removed, for example. We've seen increase in insulin sensitivity also. Normalization of brain chemistry, optimization of the metabolic pathways. We've seen tissue repair in many different areas, and the optimization of cell functions. So I believe that redox signaling and this research needs to be done in more detail. Cardiovascular research that is ongoing shows that, in fact, we did most of our studies on the endothelial cells in the veins, and we showed that in the cardiovascular system, redox signaling is very efficient in repair and replacing the damaged cells. This is backed up by research. In the endocrine system, we've also shown that increased hormonal reception is able, that insulin-producing beta cells become more efficient in a reduced oxidative stress environment. Skin research I've showed you. Lung research also has been done in endothelial cells and increased antioxidant action, and this is where we also found that the increase of glutathione peroxidase levels also were performed in these lung tissue. I'll give you these references if you're interested after the conference. Okay? Digestive, nervous system research, and all indications show that by use of clinical supplementation of these reactive oxygen species, these redox signaling molecules, that we're able to then have a significant enhancement of our ability to restore our health. So just showing you that there's some anti-aging benefits also. Here, I'll go through and then conclude with this. Because the redox signaling process is universal and fundamental to all cellular processes in animals, in plants, in every form of life that's on Earth, I believe that this redox signaling technology will advance medical science this century and beyond. Imagine being able to accelerate cellular regeneration cycles, for example. I have one thing that I wanted to say also. My wife is in the audience. I appreciate her. She cut her finger. This is when I didn't understand whether these things were possible or not. Theoretically, I understood that if we increase redox signaling potential or efficacy, we could have increased cellular regeneration. And she cut her finger, and I said, "Well, honey, could you put your finger inside a little glass of this. She put her finger inside. The bleeding stopped immediately. I'd never seen anything like this before. In under an hour, the lesion was closed. In under 1 day, there was no scarring and no indication that there had been a cut. So I was able to see healing in seconds that normally takes minutes, and healing in minutes that usually takes hours, and healing in a day that normally takes weeks. And so that was when I realized that we had truly made a very important discovery. And so I leave you with this. Thank you very much for your time.